So let's take a high-level overview of what is the substrate cycle for glycogen metabolism, which is sort of a three-part sequence for glycogen breakdown and then a three-part sequence for glycogen synthesis. So we're going to continue just with our cartoon structure right here to help us think about how we mobilize glucose in times of need, whether from the liver for global use or skeletal muscle for local use, right? And part of the key here is why we have branching in glycogen is we've created lots of these non-reducing ends, and this is where the glycogen breakdown process starts. So the first step that we're going to think about is what we call mobilization. And I want you to think about, and only because I'm going to be using this as an analogy, I want you to think about Pac-Man, right? This is my Pac-Man enzyme. It's going to be chewing in on these non-reducing ends. So this enzyme has to have an end of a molecule to chew in from. So if we imagine if we had 20,000 glucose residues that were all linked just by alpha 1,4 glycosidic bonds and no branches, we'd have one end. That would be a very slow process to mobilize that glucose. So the more ends that we have, the faster we can mobilize glucose. So the name of that Pac-Man enzyme is phosphorylase. Okay, this is also the rate determining step of our enzymatic pathway. So we are going to control flux through glycogen breakdown, which we also call glycogenolysis, with phosphorylase. Here's the chemistry that it performs. It's going to cleave alpha 1,4 glycosidic linkages, and the product from that is glucose 1-phosphate. So remember when we looked at the structure of glucose, the majority of our residues are kind of these yellow residues. So when we're breaking those down, we're generating glucose 1-phosphate. Okay, now here's the problem with Pac-Man. Pac-Man can only chew in until there are 4 residues left before you reach a branch point. It just gets too crowded. So at that point, Pac-Man has to stop and he has to wait for the next guy to sort of come in and do its chemistry. So the next step is operated by an enzyme that actually has 2 functions. We call it a bifunctional enzyme, but the purpose is ultimately debranching. So the enzyme's name is, not surprisingly, glycogen debranching enzyme. This is the enzyme name, glycogen debranching enzyme. And I want to highlight that this is a bifunctional enzyme. Now, this is the first time we've talked about bifunctional enzymes, but it is not by far, by any chance, the last time that we will see it in biochemistry. Lots of enzymes sort of have multiple stores, if you will. The analogy we used in class is imagine going to the pizza shop that also rents movies, right? It's a 2-stop shop. So here's the things that this enzyme does. The first thing that it's going to do is it's going to move short branches. Okay, so what's going to happen is Pac-Man has chewed in and basically this is what's left. It's going to take a portion of that and move it to the end of another short branch to create a longer branch that Pac-Man can chew in on again. Okay, so it moves those short branches, but the chemistry that it does to do that is going to leave a little nub right above where the branch point is. So it has a special then chemistry that's going to break that alpha 1,6 linkage and break that nub off. Okay, so the second step is it's going to break alpha 1,6 linkage. So it's going to break that alpha 1,6 linkage, and I'm going to call it on the nub. And this is actually how we get our 10% that was free glucose. So if you've already watched the glycogen structure picture, we've talked about that the number of the number of branch point glucose residues that we have, we sort of said those become glucose. Those actually don't directly become glucose. It's the nubs that it's attached to, but it's a 1-to-1 sort of relationship, so we can count the number of branch points we have, and that's the amount of free glucose that we will get. Okay, so Pac-Man has chewed in. We get so that there's too short of a branch. We're going to transfer that branch to the end of a longer branch. Pac-Man's going to chew in. Okay, so let's kind of take stock of what we have now. At this point, we're going to have about 10% is going to be free glucose, but about 90% is going to be G1P. And highlighting, that's because we got it from here. So G1P is made by our Pac-Man, our phosphorylase enzyme, and so we're going to have G1P, but that's not a branch point metabolite. So the last step is basically taking G1P and converting it into G6P, okay? And that is done by that mutase enzyme. We've seen those enzymes with mutase names before. They move entire functional groups, okay? So summarizing, we're going to chew in from our non-reducing ends until we get too crowded. Next team comes in, moves those branches, creates longer branches for Pac-Man to chew in again. Once we've done that, so that we're pretty much down to a pile of rubble, we've got 10% of that rubble is free glucose, 90% of that rubble is glucose 1-phosphate. The last partner to come in is then our mutase enzyme, phosphoglucomutase. I guess I didn't write that name down. Phosphoglucomutase. Okay, that enzyme is going to come in and it's going to convert that 90% pile of G1P into the important branch point metabolite G6P. That's glycogen breakdown. Let's talk about the flip side then. Let's say that we want to go and now synthesize glycogen. Well, let's talk about why and when we'd have that. We are going to be undergoing glycogen synthesis when we are in a fed state, which means we are going to have excess glucose. Going through the first step of glycolysis, hexokinase is going to convert that into G6P, and we can reverse that mutase enzyme that we just learned about to ultimately generate the G1P product that we need. So, big picture to highlight what's going to go on here. We've got a monomer that we need to polymerize. Monomers don't like sticking themselves together. So what we need to do is we need to activate a monomer so that it's conducive to doing that coupling reaction. And if that's all we did in that polymerization step, we'd be left with, again, 20,000 residues with one non-reducing end and one reducing end. So the last step then is to add in branches. So let's kind of talk about those steps and how they work here. First step is activation of our monomer. So the enzyme for that is called UDP glucose pyrophosphorylase. I know that's a mouthful. UDP glucose pyrophosphorylase. Okay, and basically that's going to take that G1P and make it into UDP glucose. So it activates these monomers so that they can be coupled together. And how this works is we've generated this UDP group, and this UDP group is a good leaving group. Okay, so that's why this is important. This is a good leaving group, right? We want to have somebody be a nucleophile, somebody be an electrophile, form a new bond. It's great if you've got something with a good leaving group. So that's why that first step happens. Second step, this is going to be sort of the important money step for this process because this is the rate determining step. Polymerization is accomplished through an enzyme called synthase, glycogen synthase, if you want to put that name in front of it. It is the rate determining step that we have for this process. It creates our alpha 1,4 linkages. Okay, and so this is going to be essentially creating an entirely linear homopolymer of alpha 1,4 linked glycosidic bonds in glucose residues. So if that's all that we did, we wouldn't be making a very effective glycogen molecule for mobilization of glucose in time of need. So the last step then is our branching. And the intent of branching is that we increase the number of non-reducing ends. So that's why that step is really important. Enzyme responsible for that, not too creatively, glycogen branching enzyme. okay? And it creates alpha-1,6 glycosidic linkages. So it creates alpha-1,6, may have said 1,4, creates alpha-1,6 linkages. And as we're going through all of this, I really want you to see how these two sort of breakdown and synthesis pathways are very complementary in the terms of the kinds of chemistry that happens. So I don't have a picture here of building glycogen, okay, but highlighting what's happening is we're activating all of these little spheres in our first step to make them likely to couple together, okay? Then we couple them all together and we're going to be left with these short pieces. Okay. Now, one of the other pieces that's going to happen is we need to introduce those branches. And so what's going to happen is the reverse of the debranching enzyme. We're basically going to take a long chain and we're going to take the end off of it, and then we're going to attach it somewhere in the middle of that chain using that. I'm going to flip to a structure here. It's going to utilize a C6 CH2OH group. See, we've got lots of those here. Any one of those could serve as a branch point. Okay, so we're going to introduce that branch. We'll talk more about the details when we learn about this enzyme, but just as a way to think about it, there's this trade-off between being able to put enough glucose in there and put enough branches in there. You want to have lots of branches because then you have rapid mobilization, but you also want to be able to put a lot of glucose in there. So just see if this makes sense. Let's imagine we got a little shrub growing in our yard that's got like all kinds of branches. If we branch too frequently, we're going to get really dense really quickly and we won't be able to grow very, very much bigger. And the same is true with glycogen. If we branch too frequently, we're going to get crowded and we can't add past kind of the first few shells. If we don't branch enough, then that's compromising the number of non-reducing ends we have. So there's kind of this balance between how many branches we put in, because we want lots, but not too many that we end up with really small particles. So nature has sort of figured out this perfectly. But in sort of a 30,000 foot view here, listing the enzyme names and the chemistry that they perform, the 3 steps for glycogen breakdown, the 3 steps for glycogen synthesis in what is our glycogen sort of homeostasis substrate cycle.